Hi, welcome to my presentation on human immunodeficiency virus, or HIV. In this short presentation, I will be discussing a few key points to help better understand the HIV virus. I will be discussing the basic structure of HIV and the importance of understanding the virus composition, modes of transportation, how the virus replicates, viral load and CD4 cells, the body's immune response and the consequences of the immune system compromise, as well as treatments for the virus, research and advancements in microbiology, the global impact the virus has had over the years, and the ongoing efforts for vaccine development. It is important to understand the basic structure of HIV and the importance of the virus's composition. Understanding the basic structure of HIV is crucial for developing antiviral drugs, vaccines, and diagnostic methods to combat and manage HIV infections. The structure of HIV is complex and consists of the following key components. The envelope, which has a HIV has a lipid envelope derived from the host cell membrane. This membrane surrounds the viral particles and contains glycoproteins, particularly GP120 and GP41, which plays crucial roles in the virus's entry into host cells. Next, we have the capsid. Inside the envelope, there is a protein shell called the capsid. The capsid is composed of P24 protein molecules. The capsid protects the viral RNA and enzymes. Next, RNA. HIV carries its genetic information in the form of RNA rather than DNA. This RNA encodes the necessary instructions for the virus to replicate and produce new viral particles. And then lastly, we have enzymes. There are three. Reverse transcriptase. This enzyme converts the viral RNA into DNA during the early stages of infection. It integrates the viral DNA into the host cell's DNA. And then protease, which facilitates the maturation of new viral particles by cleaving large precursor proteins into functional components. The next slide shows a picture of viral replication. We will be discussing the steps of how the virus replicates. Viral replication involves several steps. Here is a simple step-by-step process for the replication of a virus like HIV. First, we have attachment. The virus attaches to specific receptors on the surface of a host cell. In the case of HIV, it typically targets CD4 receptors on T cells. Then we have entry. The virus enters the host cell. HIV, for example, fuses its envelopes with the host cell membrane, releasing its contents into the host cell. Next, we have reverse transcription. In the case of retroviruses like HIV, the viral RNA is reverse transcribed into DNA by the enzyme reverse transcriptase. The viral DNA is then integrated into the host cell's DNA. Transcription and translation. The integrated viral DNA is transcribed and translated by the host cell machinery to produce viral RNA and proteins. Then assembly. Newly synthesized viral RNA, proteins, and enzymes move to the cell membrane and assemble into new virus particles. Following that is budding. The newly formed virus bud off from the host cell, acquiring an envelope derived from the host cell membrane. This process can often leads to the destruction of the host cell. And then finally, we have maturation. The newly released virus particles undergo maturation involving the cleavage of precursor proteins by the viral protease, resulting in the formation of mature infectious viruses. Understanding these steps is crucial for developing antiviral drugs that target specific stages of viral replication cycle, aiming to inhibit the virus's ability to infect and replicate within host cells. Next, we will discuss the transmission of the virus. There are several ways that the virus can be transmitted. Here are some common ways. Unprotected sexual contact. Transmission can occur through unprotected vaginal, anal, or oral sex with an infected partner, especially if there are open sores, cuts, or mucous membrane exposure. Mother-to-child transmission. HIV can be passed from an infected mother to the child during childbirth, through breastfeeding, and during pregnancy. Contaminated blood products. Transmission can occur through the use of contaminated needles or blood products, such as intravenous drug use, or unsafe medical practices. Occupational exposure. Healthcare workers can be at risk if they come into contact with HIV-infected blood or bodily fluids through accidental needle sticks or other occupational incidents. Sharing needles or syringes. IV drug users who share needles or syringes are at an increased risk of HIV infection. Organ transplants. In some cases, if organs from HIV-infected donor are transplanted without appropriate precautions, there is a risk of transmission. Understanding these transmission routes is crucial for implementing preventative measures and promoting safer practices to reduce the risks of HIV. Next, we will discuss viral load and CD4 cells. Viral load refers to the amount of virus, specifically the concentration of viral particles in a person's blood or other bodily fluids. In the context of HIV, it typically represents the quantity of HIV RNA present in a blood sample. Measuring the viral load is crucial in managing HIV or AIDS because it provides information about the intensity of viral replication within the body. A higher viral load generally indicates more active viral replication and can correlate with a faster progression of the disease. So monitoring the viral load serves several purposes. The first is assessing the disease's progression. Increasing viral load may indicate a progression of HIV infection and a decline in the immune system's ability to control the virus. Next, we have evaluating treatment effectiveness. So viral load testing helps assess the effectiveness of antiviral therapy. A decreasing viral load indicates that the treatment is suppressing the viral replication. And then predicting transmission. Individuals with a higher viral load are more likely to transmit the virus to others. Effective treatment can reduce the risk. Regular viral load measurements, along with CD4 cell count monitoring, are crucial components of HIV care. The goal of HIV treatment is often to achieve and maintain an undetectable viral load, meaning that the amount of the virus in the blood is so slow that it cannot be reliably measured by standard tests. The impact on CD4 cells and immune function. HIV has a profound impact on CD4 T cells, a crucial component of the immune system. The virus specifically targets and affects these cells, leading to several significant consequences. There is CD4 cell destruction, impaired immune response, immunodeficiency, CD count as a marker, and impact on adaptability immunity. In the next slide, we will discuss the immune response to HIV. HIV specifically targets and affects the immune system, which plays a crucial role in developing the body's defense against infections. Here's how HIV impacts the immune system. First, HIV primarily targets CD4 T cells, which are a type of white blood cells that coordinate immune responses. Next, cell damage. HIV actively replicates within CD4 cells and T cells, leading to their destruction. HIV leads to chronic immune activation as the immune system attempts to respond to the ongoing viral replication. Losses of immune coordination. The decline of CD4 and T cells disrupts the immune system's ability to coordinate responses to infection and other immune challenges. Impaired antigen representation. Antigen-presenting cells, such as dendrite cells and macrophages, are also affected by HIV, impacting their ability to present antigens to CD4 and T cells. Reduced B cell activation. CD4 and T cells play a crucial role in activating B cells, which produce antibodies. The loss of CD4 and T cells hampers this activation, leading to reduced antibody production. Decreased cytotoxic T lymphocytes function, which are essential for recognizing and eliminating cells infected with viruses. HIV can repair the function of these cells, reducing their ability to control the virus. And then finally, immune suppression and immunodeficiency. Progressive loss of CD4 and T cells eventually result in immunodeficiency, where the immune system becomes severely compromised. Immunodeficiency increases susceptibility to opportunist infections and certain cancers that a healthy immune system would typically control. The impact of HIV on the immune system is dynamic, involving a complex interplay of virulent replication, immune responses, and the gradual loss of key immune cells. We will now discuss the impact that HIV has had on a global level. In 2020, approximately 37.7 million people were living with HIV globally. Africa remains the most affected region, accounting for the majority of global HIV cases. In 2020, again, there was also an estimated 1.5 million new HIV cases worldwide. The global number of AIDS-related deaths in 2020 was around 680,000. And then Africa is the most heavily affected region, with the highest prevalence rates and the majority of new infections and AIDS-related deaths. Other regions such as Asia and the Pacific, Latin America, the Caribbean, Eastern Europe, and Central Asia also have varying levels of high prevalence. Young people, ages 15 to 24, accounted for a significant proportion of the new HIV infections globally. In several regions, there were concerns about HIV transmission among key populations, including men who have sex with men, sex workers, and people who inject drugs, as well as transgender individuals. I have included a pie chart showing here new HIV infections as of 2021, and it's separated into the different global population groups. Research and advancements. So we'll discuss some research that has been done and some of the breakthroughs that they've had. So long-acting antiviral drugs. They continue to research the formula for antiviral drugs, offering alternatives to oral, daily medications, injectables, and implantable options. HIV vaccines. There's ongoing research on HIV vaccine candidates aiming to stimulate effective immune responses against the virus. Different vaccine approaches, including mRNA and viral vector vaccines, are under investigation. Developing and testing of broadly neutralizing antibodies, which have the potential to target a wide range of HIV strains. These antibodies could be used for both treatment and prevention. PrEP, which is pre-exposed prophylaxis. So these are used for anyone that may have an increase in risk behavior of contracting HIV. It helps with the transmission. Dual antiviral therapies. So this is basically exploration of two different types of therapy regimens involving the use of two drugs instead of the traditional three-drug combination. And it aims to reduce side effects. Gene editing technologies. Advances in gene editing technologies for potential HIV cure strategies. It's an ongoing research to help explore approaches to eliminate and suppress the HIV virus within the human body. And then cure research, ongoing investigations into strategies for achieving a functional and complete cure for HIV, aiming to eliminate viral reservoirs and achieve long-term remission. So let's talk about prevention strategies. Condom use. Consistent and correct use of condoms during sexual activity reduces your risk of HIV transmission. PrEP, as we spoke about on the last slide, taking antiviral drugs before potential exposure can be effective in preventing HIV infection, particularly those that are at high risk. Regular HIV testing. Knowing one's HIV status through regular testing enables early detection and timely intervention. Needle exchange programs, providing clean needles and syringes to IV drug users helps reduce the risk of HIV transmission. Antiviral treatment, or ART, for individuals with HIV, adhering to an antiviral therapy can suppress the viral load, reducing the risk of transmission to others. Safe practices during pregnancy. Proper medical care and antiviral drugs during pregnancy can significantly reduce the risk of mother-to-child transmission. And then finally, awareness and advocacy. There are several organizations that help make people aware and advocate for people that are infected with the HIV virus. There's World AIDS Day. It's an annual observation of World AIDS Day. It's on December 1st to raise awareness, commemorate those infected with HIV or AIDS, and promote advocacy. There's the Global Fund. It supports international initiatives like Global Fund to fight AIDS, tuberculosis, malaria, and the U.S. President Emergency Plan for AIDS Relief. Community involvement, engagement of non-governmental organizations and community-based organizations for outreach, education, and advocacy. Stigma reduction, efforts to reduce HIV-related stigma and discrimination, creating an environment that encourages testing and treatment. And then again, research and development continues to be ongoing for new prevention methods, treatment options, and potential cures for HIV. Thank you for listening.